Genetics Lesson 2.7 Sex-linked traits. So, what are sex-linked traits? So they're multifactorial, which means they're traits whose phenotypic expression is controlled by the genes found at the different loci. So if you remember all the way back into the DNA lesson, we talked about the loci, and that's the place, that sort of center where they meet at. And then linkage means that when genes occur at the same chromosome, and then we have to remember that each chromosome has hundreds of different types of genes. And so the expression is influenced by internal and external environmental conditions. So when we think about things like our height, that's something that is controlled. People can say, well, your parents, one of your parents is like 6 foot and the other one is 5'9", you're obviously going to be tall. But then there's other people whose parents are really short and they just ate different types of foods, or they did different sports that made them really tall. So that's what we call the nature versus nurture. So nature is your genes and your genetics and like the science behind how tall you are. But then there's also a bit of nurture in it, where did you get enough exercise as a kid? What kinds of foods were you eating? So there's also that nurture aspect in there as well. And different ways we can study this is through examples like identical or monozygotic twins, because they have the exact same DNA. So how is it possible that they could be different heights? Or how is it possible that they could like different things? Because they're supposed to be genetically the same. So scientists use monozygotic twins to help understand the effects of nature versus nurture. Okay, so let's talk about autosomal versus sex-linked. So autosomal means that it's carried on any traits that do not determine gender. Sex-linked is a trait or disease carried on the X or the Y chromosome. So we have to remember that a sperm is either X or Y, and the egg is an X. So males are X and Y, and females are XX. So when the sperm and the egg come together, the baby could either have these two and be a female, or the baby could have these two and be a male. The way that we determine the sex is by the 23rd chromosome, so whether that is an X or Y. And sex-linked diseases, which is what we're learning about right now, are always carried on the X. So if we'll use red for a girl and blue for a boy. So if we have XX, which is a girl, and we have XY, which is a boy, the trait is always carried on the X. And the way that we show that is with the X with a little H, or whatever the letter is, on the X. So if I were to show this girl here, are both her X's affected? No, she only has one X that affected. So therefore she is a carrier. But what if she looked like this? Both her X's are now affected, so that means that she does in fact have the trait now. Because both her X's are affected, they both have that little H on it. The boy, however, they cannot be carriers. So remember, it can only be carried on the X, so only girls can be carriers. Because if I put this little H here, because remember, it can only be attached to the X, this boy has it. Because it doesn't have another X to spare in order for it to be a carrier. He just automatically has it. So hopefully that gives you a better understanding of how we determine the sex-linked traits. So now let's do a quick problem practice. So in the human, the gene for normal blood clotting is dominant to the gene that causes hemophilia. The trait is X-linked, so it already tells us that it's X-linked. A woman who is a carrier of hemophilia is married to a man who has normal blood clotting. What are the chances that they will produce a hemophilic son? So it tells us, well, first we should write that H is going to equal the trait for hemophilia. It tells us that the woman is a carrier. So if you remember how we write a carrier, we just infect one of her X's. If we did this, then she would have the disease, but she's just a carrier right now. And it tells us that the guy doesn't even have it, so he is just regular. Now we can do our Punnett square to cross between those. So remember, we show our cross between this, and we write our Punnett square out. And always remember that what the question is asking you. So we just write this out regularly, except we take the H with us. So this one would still be a normal cross, XX. This would be XY. And then this one, we just take that little H with us. So when we write this, we write it there. Usually we write it on the first H, but it doesn't matter. On the first X, sorry, but it doesn't matter. And this one will be XHY. So you just take it with you. And just to make it easier for you to see, this one is a girl, this one is a girl, this one is a boy, this one is a boy. So it asked us, what are the chances that they will produce a hemophilic son? So we're just looking at the blue ones in this case. Well, obviously this one right here, it doesn't have the H on it, so that means that he does not have it if they were to have that combination. However, they do have that one who could have hemophilia. So that means that their son has a 50% chance of having hemophilia. Okay, so now we're going to talk about pedigrees. So pedigrees are little family trees or diagrams, if you will, that help us determine if people are carriers of a disease that runs in the family, or if the children are going to have that disease. So on the side here are just like the key or the symbols that we use to write these out. So males are written as squares and females are written as circles. When they're married or they're together, you draw a line between the two, just like I have drawn out on the side, and then we draw the children in birth order. Those are really the main things that you need to know. And then on the side up there, there's affected individuals, which we color in. And then if it's autosomal recessive, we'll talk about that in a second. You shade it halfway, and if it's carrier for a sex-linked recessive, then you draw a dot in the middle. But we'll talk about that in a second. Here's just a really simple pedigree on the side here for a family who has the trait for colorblindness. So I have already shown you that the mom is a carrier for the trait. So the way we would write her is XCX, because one of her C's is infected. She doesn't have the full disease. She's not like this. She just is a carrier for it. She is, however, married to a man who does have the disease. Same with her son and a daughter whose both X's are infected by the disease. So although the mom has normal vision, she has to be a carrier because she has a daughter with the disease. Because the daughter would get one X from her mom, and she would get one X from the dad. And this daughter's just so happened to get both bad X's from both parents. Now, if we look at the daughter over here, if she got both, there is a possibility for this daughter over here to be a carrier of the disease. Since she doesn't have it, we know that she doesn't have it because she's not colored in, she could very well be a carrier for the disease. So we add a little circle like her mom over there. And then this last brother over here will just be plain XY because he gets one good Y from the dad. He gets his Y from the dad. He gets the good X from the mom, which makes him not have the disease, obviously, because you get your X's from your mom. And essentially that's how you draw a pedigree. Here's just what I was talking about before with the autosomal. So autosomal means that more girls have the trait, so that means more circles are colored in. Sex-linked or X-linked means that more boys have the trait, so that means that more squares are going to be colored in. And then you'll see words like recessive and dominant. So recessive means that in the sort of like pedigree or the chart, it skips a generation. So here you'll see that this has skipped this entire generation. No one in this line has the disease, so that means that it will be called recessive. But if it's dominant, that means that every generation would have at least one person to have the disease. So this picture down here would be sex-linked or X-linked, just they're interchangeable, recessive, because it did skip a generation and more boys are colored in than girls. Now, just before we end the lesson, I'm going to do a quick thing on blood types. They're not really connected to sex-linked or X-linked or autosomal or anything. It's just a good thing to understand because you might need to do pedigrees for these. So I'll just write out the alleles for blood types. So alleles, phenotype, and genotype. So over here we have IA, and its phenotype is going to be blood type A, which some people have blood type A. It could have two different genotypes, so it could either be IA little I or just big I with an A. And that's because this one over here could have had the recessive trait in it. This one is going to be IB, and you guessed it, its blood type is going to be blood type B. Now it could be IBI or I like that. And the last one is just this little I, just like we saw over here, and that is going to be representative of blood type O, and it could be II. That's the only one that blood type O could be. The phenotype for blood AB, because some people do have blood type AB, the only one that it could be was IAIB. So you could get pedigree problems with some of these in it, and they're super easy to answer. All you have to do is determine what the genotype would be for this person with that blood type. So if the child had, let's say, blood type O, II, and the parents were A and B, the only way that it could give the child these two little I's here to make it this is if the parents were big I, little I, big I, little I. In these ones over here, there's no little I's, so the child could not be blood type O. So that's sort of how you have to go about answering those types of questions. Other times it's super easy and it's easy to figure out, and you just have to make a simple pedigree to determine what the child could be. And that's it for sex-linked diseases and traits and blood types, also pedigrees.